So now we've talked about the whole process, right? We've gone from how light works. We've talked about the whole process of photosynthesis, summary of photosynthesis. We've talked about the light reactions. We've talked about the dark reactions. So we should be done, but we're not. Because carbon fixation is very important to plants, and they need to be able to do it in every possible environment. The reactions I just told you about have some serious drawbacks. They have some serious disadvantages in hot, arid environments. So what I want to do now is go through ways that plants have adapted to living in hot conditions. The name of the game is reproduction. If you reproduce the most, you're able to send the next generation the best, most beneficial alleles. I used words you might not know. You send the next generation the traits that you have that allowed you to survive. Whether it makes, spiky flowers, for instance, might allow you to survive better than soft, delicious flowers. So plants that are in hot environments had to adapt new ways to deal with all of the heat and all of the water loss. What we have just learned is what's called C3 photosynthesis. This is the one you've been learning your whole life. This is the one that I just went over with you for a few hours. But it's not the end-all be-all. C3 photosynthesis starts with a 3-carbon molecule. I emphasized that in a previous lecture. 1, 2, 3 carbons, and that's where it gets its name, C carbon 3. You see examples of this all over the place. Oak trees are C3 plants. Soybeans, strawberries, potatoes, wheat, all of these are C3 plants, and you'll notice they all require a ton of water. See, C3 plants love cool, moist environments. When are strawberries in season? In the summer? Is it summer? I thought it was spring.
It's like
beginning of summer. But you'll notice what's always set up at the strawberry field? Lots of sprinklers, right? You got to keep giving those things tons of water. They love water. These are hellebore plants. Hellebore plants grow in the shade, and they need lots and lots of water. Water is the big limiting factor. In these C3 plants, photosynthesis takes place throughout the entire leaf, except the epidermis, all the cells of the mesophyll. There's lots and lots of photosynthesis going on. Aside from the chloroplast, they don't need any special anatomy. They just need chloroplasts in them with some chlorophyll and the thylakoid membrane, and they're fine. But the problem with C3 plants is they are very, very bad at using their water efficiently. The problem is this plant will keep its stoma open for a long time. And when the stoma is open, what leaks out? Water. What's that process called? Transpiration. So, it's just right here, yeah. So water leaks out, but it's able to absorb carbon dioxide. In an area, though, where there's lots and lots of water, this isn't a problem. Water comes in, and then it gets just dropped immediately. But that's okay, there's always more water. When there's a lot of water present, this is not an issue at all. These guys just keep going. In C3 photosynthesis, you've got rubisco. Again, what's rubisco? It's a very abundant protein, but the big thing is it's an enzyme. Its job is to take carbon dioxide and attach it to RUBP. In C3 plants, rubisco is doing two things. And I want you to sort of emphasize that these are two distinct processes. Process 1, over here. Take carbon dioxide out of the atmosphere. That's all it does, take it out of the atmosphere. Process number 2, attach carbon dioxide to RUBP. See how those are two distinct things? One is I can grab the carbon dioxide. The second process is I attach that to RUBP. So far, so good? Questions on that? Two distinct things going on. Carbon dioxide out of the atmosphere and attach carbon dioxide to RUBP. There's an issue with that, though. As temperatures go up, what happens to an enzyme? It starts to lose its efficiency because of what? Denaturation. So you start to lose the shape of the active site. The first thing that happens to Rubisco when it loses the shape of its active site is it starts to grab oxygen instead of carbon dioxide. That's called photorespiration. You grab oxygen and you stick the oxygen on the RUBP. Well, at that point, it goes through a whole new process, photorespiration. Photorespiration is complicated. I put up one slide of it. I put this up only to show you what's going on. Up there, you see that it says 1. See the 1? 1 ribulose bisphosphate. It attaches to the O2. Then you end up producing these secondary molecules. All right, now you've got an oxygen attached to it. What's the organelle that's used to remove oxygen? The peroxisome. So it moves from the chloroplast to the peroxisome. Then this is fun. From the peroxisome to the mitochondrion. From the mitochondrion back to the peroxisome. You'll notice that you're taking NADH and converting it to NAD+, because you're using some energy to do that. Over there too. And then you're going up there and you're converting ATP to ADP. And the whole thing, all you're doing is recovering your original particle. You're spending energy to pretty much get right back to where you started. That's what photorespiration is. Okay, let me put it in terms you might understand. Have you guys ever done a homework? You got an assignment, you look at the instructions, right? I assume you look at instructions before you start an assignment. That might be a bad assumption, but you've been there, right? Have you ever written an assignment, then after you were finished, look at the assignment and realize you did the entire thing wrong? Yeah, that sucks, doesn't it? That's like the worst feeling in the world. So what do you do after you realize it's wrong? You just say, to heck with it, turn it in. You do it again. That's what I would suggest, to maintain a grade. But, you be you. The idea is you did all this work, you put a lot of energy into it, only to end up right back where you started. That's like photorespiration. A lot of energy invested, not much, no return. You guys, a lot of you are going to go and apply to a 4-year school. The way I was thinking about this is as a transfer student, right? You're going to look at that 4-year school and you have to fill out the right paperwork, and that can be daunting because there's a lot of it. You got to fill out a form that says where you're going to go and what process you're going to go to get there. If you fill out the wrong form, it might set you back a ways. You might not get everything in on time. You may not be able to start for an extra year or an extra semester. Like with you guys, there's a difference between guaranteed articulation and transfer. If you go with one, they automatically let you in, no more questions. With the other, if you fill in the wrong piece of paper, there are going to be problems. You might not get in. Photorespiration is, you could think of Rubisco almost like that student. They've got a lot of options in front of them, and sometimes they choose the wrong one. And when they choose the wrong one, it sets them back a little. All right, so those are C3 plants and the problems with C3 plants. The big problems are at high oxygen levels, that Rubisco grabs oxygen. At high temperatures, Rubisco grabs oxygen. When there's not enough water in the soil, you can't keep the stoma open for very long. So again, photorespiration. This led to what's an adaptation called C4 photosynthesis, and this literally changed the entire environment of the planet when it came about. It made the Earth cool down pretty quickly. The first organic molecule created is a C4. It's got 1, 2, 3, 4 carbon to it. Examples might include corn or Bermuda grass or sugarcane. You guys ever looked at your lawn before? I assume you have. You look at your lawn when you get a chance. You'll notice there's grass on the ground, probably, right? Okay, welcome to Virginia now where it's all mud and moss. But there used to be grass there. If you notice, there are 2 types of grass in your yard. There are grasses that are growing under trees that are very soft, and then grasses that are growing in full sun that are very spiky. You notice that? Walking barefoot in your yard, there are patches that you walk on, and there are patches you don't. The stuff growing under the trees are C3 plants, cool, moist environments. The ones growing out in the sun are likely C4. These C4 plants do really well in hot, dry regions. The big thing to note is that photosynthesis takes place in the inner cells of the mesophyll, not throughout. So this is a cross-section of a leaf of corn. You'll notice that there are these outer cells that surround this inner circle. Photosynthesis happens inside that circle, not these outer ones, only the inner ones. It has this specialized anatomy. That circle is called the Kranz anatomy. It means wreath in German because it's shaped like a wreath. The way that the C4 photosynthesis has a huge advantage over C3 photosynthesis is its ability to bind CO2 efficiently. Carbon dioxide comes in through what structure in the bottom of the leaf? What's it called? The stoma. So carbon dioxide comes in. Now, photosynthesis doesn't happen here, but they have this molecule called PEP carboxylase, PEP. That PEP carboxylase will bind only to carbon dioxide. It will never, ever, 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 ever bond to oxygen. No matter how hot it gets, it will only bond to carbon dioxide. So that PEP carboxylase then takes the it takes the carbon dioxide over to the inner cells. So it goes from here, the stoma, across the cell to the inner cell. It's carrying with it carbon dioxide. When it gets to the inner cells, it drops off the carbon dioxide, where it hands it off to Rubisco. So now Rubisco, no matter how hot it gets, doesn't have any option but to choose carbon dioxide. It's sort of like, I don't know if you've ever tried to make somebody make a decision that you wanted them to make, so you put the two things you wanted them to decide in front of them. I was shopping with my son yesterday, which is never fun. He had to get a pair of pants, and he didn't want pants, so I picked out two pairs of pants that were exactly the same in every regard, and I held them up and I said, "Which one of these two do you want?" And he pointed, and that's the one we got. He had a choice, right? It's just the choice was exactly what I laid out in front of him. That's what the PEP carboxylase is doing. It's dropping off carbon dioxide to the Rubisco. Now the Rubisco can only choose carbon dioxide. Let me ask you, do you guys remember what these holes are between plant cells that allow them to pass nutrients and chemicals back and forth? Starts with a P. Ends with a plasmodesmata. Plasmodesmata, good, okay. Plasmodesmata. So you're moving carbon dioxide in, the PEP is grabbing it, taking it across into the inner cells of the mitochondria, into the inner cells of the Kranz anatomy, the mesophyll, where they can power the Calvin-Benson cycle. So Rubisco can now take the CO2, attach it to RUBP, and the process begins. So for you guys, as a transfer student, it'd work out really well for you if you had somebody sitting in front of you whose whole job it was to say, this is the paper you need to fill out, right? Tax season's coming up. Be really nice to have for those of you who do taxes, really nice to have somebody sitting next to you saying, this is the form you fill out. Good. All right, now this form. Now this form. It makes things easier. That's what PEP carboxylase does. Make sure the Rubisco always gets the right material. So that's C3 and C4 plants. Where C3 plants, the first molecule is 3 carbon, and C4 it's 4 carbon. C3 is in cool, moist environments. C4 is in hot, dry environments. C3 photosynthesis is throughout the leaf. C4, it's in the inner cells of the mesophyll. C3, you don't need any specialized anatomy. C4, you need that Kranz anatomy. C3, rubisco pulls CO2 from the environment, sometimes oxygen. In C4, it's PEP carboxylase that only pulls CO2 from the environment and takes it to the rubisco. But wait, there's one more. You might ask yourself, what about pineapples? Because that's what I always ask. What about the pineapple? First off, I want to point out pineapples don't come from trees. This is what pineapples grow on. In case you didn't know, it's super cool. They grow up on a stalk, and then you just sort of break them off. The more you know. Pineapples, cacti, aloe, that substance now you're all questioning, and other succulent plants only open their stoma at night. They live in super hot, dry conditions. If they open their stoma during the day, lots of water will evaporate out. So instead, they only open up at night. So by opening up just at night, they're able to retain water on the insides. The problem is that's the only time they can get carbon dioxide. They cannot pull in carbon dioxide during the day. What do they use that carbon dioxide for? Not to make water. What's the whole point of photosynthesis? Make glucose. So if they can't pull in carbon dioxide, they can't make glucose. So they got around this. The way they get around this is at night, the stoma open up. They suck in a lot of carbon dioxide. They stay open for a long time. But they don't use all the carbon dioxide. They take that carbon dioxide and they turn it into malic acid. That malic acid, it's an organic acid. That's not important. The big thing is they turn it into something else. And then daylight comes, and they close up. The stoma shut immediately. Throughout the day, they can take that malic acid that they've built up overnight and break the carbon dioxide out of it. So it's sort of like storing something in a fridge for later. So they've stored carbon dioxide as another molecule. Then during the day, they break down that molecule so they have a constant supply of carbon dioxide. So they can constantly make sugar. So again, that's called CAM photosynthesis. Now that both CAM and C4 photosynthesis occur in hot, dry conditions, the big difference is C4 photosynthesis is going to require some very specialized anatomy. That Kranz anatomy, it requires 2 cells, whereas CAM photosynthesis only requires 1 cell. C4, the stoma will open or close day or night, but in CAM, the stoma will only open at night. So do you guys have questions? Where did we end? We'll come back to the front row. So we talked a lot about C3, C4, and CAM plants in this mini lecture. C3 plants, C3 is the photosynthetic mechanism that we talked about at the very beginning of this group of slides. It's sort of the model that we used. And then we looked at variations with C4. C3 plants don't do terribly well in dry or hot environments. They have a relatively poor water use efficiency. So you'll see a lot of C3 plants growing in cool, moist areas, maybe under other trees or near a creek. C4 plants, however, they grow in hot conditions. They grow when in straight up sunlight. You'll see these in the middle of your yard where there's not really a lot of water. The reason C4 plants are so much more efficient is because C3 plants use rubisco to do 2 things: pull carbon dioxide out of the atmosphere and attach it to RUBP. C4 plants, on the other hand, use PEP carboxylase to grab carbon dioxide from the atmosphere, and then rubisco's only job is to take the carbon dioxide from PEP and attach it to RUBP. It does that by living in the center of the leaf in the structure known as the Kranz anatomy. So that lets plants keep their stoma open for less time, because they're so much more efficient. As the stoma are open less, they lose less water to the environment. CAM plants are sort of hyper good at surviving in hot, dry conditions. CAM plants are plants like pineapples, like cacti, like aloe, like succulents. These plants keep their stoma closed all day long. They never open up to take a breath of carbon dioxide. Instead, they only open at night, and they allow carbon dioxide to come in at night, and they turn that carbon dioxide into an organic acid. Then during the day, the stoma are closed, and the plant breaks down those acids all day long in order to release carbon dioxide within the plant that is then used to produce sugar. So it's hyper efficient in desert conditions, but not terribly good in wet conditions. These content review questions are there to really focus your studies. Hopefully now we've got an understanding of not just cellular pathways or metabolic pathways, but also examples of how those metabolic pathways can work. The next group of mini lectures is going to move on to a new topic. We're going to start talking about how information is passed from one generation to the next. So to start with, we're going to briefly discuss the cell's life cycle and talk about how the cell can reproduce itself in a process known as mitotic cell division. And then talk a little bit more about meiosis and mitosis. So coming up next, the cell cycle.